introduce Laura Wasylenki and to welcome her back to the department. In fact, Laura was a visitor in this department for a couple summers, quite a number of years ago. She's a really old friend of mine because she and I worked together in the same research group when we were at Caltech. I was a postdoc and she was a graduate student, which indeed means that she started out as an experimental petrologist. And we worked together closely enough to actually co-author a paper. But I think the one thing that Laura knew when she finished at Caltech was that she didn't want to do research in experimental petrology anymore. And so she's had a career path that's very interesting, and I think that's worthwhile as a counterexample, as a very positive counterexample to our more conventional path, which is that we assume that people end up in research because they did a PhD in a certain direction, and then they put one foot in front of the other and then ended up where they were. And Laura has not put one foot in front of another. She's taken some alternative paths that were
I ran away.
And it has worked out for her. So like I said, she knew she didn't want to do research in experimental petrology. So after her PhD, she took a job at a small college in New York teaching. And after a few years realized that she really missed research. It's not that she missed experimental petrology, but that she missed research. And she did something extraordinarily brave, which is that she gave up that faculty position to start again and to become a postdoc. And she worked for a number of years as a postdoc with Patricia Dove at Virginia Tech, doing experimental studies of biomineralization. And that was more in the right direction for her. And then after that, she moved sideways and became a research scientist at Arizona State, working with Ariel Anbar, where she worked on fractionation of metal isotopes during mineralization, including biomineralization. And that has worked very, very well for her. And she's made a number of really important studies, both of metal fractionation in inorganic and biogenic environments, modern environments, and ancient environments. Following her time at Arizona State, she took a faculty position at Indiana University, where she established a mass spectrometry lab. But Laura is a dyed-in-the-wool Southwesterner and had the opportunity to move to Northern Arizona University a number of years ago, which is where she is now. And she's the leader. Actually, I'm going to forget the acronym, but I think it's on her first slide. She's the leader of the Sesame Lab, which is basically her research. And her topic today has to do with the rise of oxygen and the role of a trace metal, nickel, may have in it. So like I said, I'm really pleased to introduce Laura. Thank you.
Great, thank you very much, Mark. Yeah, for everyone in the audience, especially the students, I want to edit what Mark said just a little bit. When I finished my PhD, it wasn't that I didn't want to do research in experimental petrology anymore. It's that my confidence was terrible. I thought that I wasn't good enough to do research in experimental petrology anymore. And so that's why I took a teaching job at first. And it was only after I went to my postdoc and then the position at Arizona State where I had different mentors who really helped me find my self-confidence and taught me how to do things like write proposals that can get funded. So for the students, I guess there are two lessons there. One is that, I don't know, just even if you don't have good self-confidence, you can still find your way towards the thing that you really want to do. And the second one is, it takes a long time for Mark to introduce me because I've had, I don't know, 6 jobs since I left my PhD, that you can always change your mind about your specialty, where you are, or what you're doing, right? That you sometimes feel like you're forced to make a pretty poorly informed decision about what grad school to go to and what topic to do your research on. You're not stuck with those things with all the other doors closed. You can always go back and reopen other doors if you want to. Sometimes it takes some hard work to climb another learning curve, but you can do it. All right, so let me switch over and share my screen and get this Zoom window off my screen. No, I don't want to leave the meeting. Okay, hopefully everyone can see that. Let me just check, there's something in chat. Let me just make sure it's not important. I don't know why the chat's not coming up. So I'm just gonna go. So I'm at Northern Arizona University, and the lab is called Systematic Experimental Study and Analysis of Metals in the Environment, or Sesame Lab. And the talk that I want to give today is about what nickel isotopes can and cannot tell us so far about Earth's midlife crisis. And I'll explain what I mean by that in just a moment. But first, let me make a very important acknowledgement that all of the data that you'll see in this talk today, and quite a bit of the thinking and the suggestion that we submit the work to Nature Geoscience, came from my former postdoc, Shuizhong Wang, who's now assistant professor at China University of Geosciences in Beijing. It was wonderful working with him, and this was a really, really fantastic project that he did. We had support from the Geobiology and Low Temperature Geochemistry program at NSF and from the Exobiology program at NASA. All right, so one theme of my group's research, it's all about trace metals, but so some of our projects, we're interested in the trace metals that are dissolved in the oceans. The ocean has sodium and chloride, of course, but it actually has a little bit of every element in it. And the amounts of those elements and their speciation and what happens to them and how long they stay in the ocean is affected by a whole bunch of complicated things, like the composition of the crust, both oceanic and continental, composition of the atmosphere, weathering rates, microbes and other organisms can often act to make change in metals, marine cycle, hydrothermal activity, precipitation of minerals, redox cycling in marine sediments, etc. So it's pretty complicated to figure out what controls the amount of different metals, especially the ones that are essential for biology. Right now, we don't even understand the modern budgets for some of the bioessential trace metals like nickel, zinc, or copper. We can't make a balanced box model or say why the amount of nickel that's in the ocean is what it is and not 1,000 times bigger or 1,000 times smaller. So it's going to be pretty darn challenging to reconstruct metal chemistry of seawater in the past, but we're interested in doing that difficult thing because we want to find out how have shifts in seawater's trace metal composition shaped the course of evolution. If some element became very scarce or became abundant, that would have had a big ecological effect. And we're interested in finding the turning points in history when trace metals may have played roles in shifting ecology and evolution in one direction or another. The other theme in my lab is about, again, trace metals, heavy metals that are toxic, and starting to learn about the reactions that control their mobility in the environment. So it's easy to go to a contaminated field site and put in a whole bunch of monitoring wells and measure some things in the pore waters and make a contour map of a contaminant. But what's really hard is predicting how much contaminant will flow how far and how fast. And the problem with that is that we can do some lab experiments and things, but we don't have any good way to determine in situ in some contaminated setting what reactions are the ones controlling some toxins' mobility. And so in our lab, we're trying to develop some new tools for doing that. Okay, but it's the first theme that I'm focused on today, and in particular looking at Earth's midlife crisis, which was 2.4 billion years ago. And so this plot here that's called the Great Oxidation Event, the plot has how much oxygen was in the atmosphere as a fraction of what it is today. So that goes from 0 to 1. It's supposed to be roughly a linear scale, but this is a highly schematic diagram. The main point is that we had the Archean eon, which went up to 2.4 billion years, when the oxygen level in the atmosphere was extremely small. And then, fairly abruptly, the amount of oxygen in the atmosphere increased to a few percent of what it is today. And we don't know too well if there were some bumps and wiggles in between. A lot of people are working on that right now. And then there was a second big rise of oxygen just before the Phanerozoic started, with the Precambrian-Cambrian boundary. So what did this great oxidation event look like? This is one of the most profound turning points in all of Earth history. And the Archean eon, before the midlife crisis, had an atmosphere that was richer in methane and richer in CO2 than what we have now, devoid of free oxygen. And we know this in part because there's a weird mass-independent sulfur isotope effect that shows up in marine sediments of this age. And so here's this water column that's supposed to symbolize a water column that's anoxic, there's no dissolved oxygen in it, sediments that are even more anoxic and somewhat sulfidic. And at the top, there are these tiny little green blobs, because it might have been the case, it probably was the case, that photosynthetic organisms to make little transient bits of oxygen, right? They would pump out a bunch of oxygen in some location where they were very productive. But these little bits of oxygen, if they did exist, would have been transient and not very extensive, right? And by contrast, after the transition, the atmosphere now had a little bit of free oxygen, less methane, less CO2, and the weird sulfur isotope effect that goes with no oxygen went away. And so in the Proterozoic eon, the top of the water column was somewhat oxygenated, and the lower water column was loaded with sulfate, which turned to sulfide, right? Because once there was oxygen in the atmosphere, sulfides in rocks could weather, delivering sulfate to the ocean, and then bacteria could reduce the sulfate and make the ocean very sulfide rich. So this was a huge, profound change. I was talking with my PhD student a couple of days ago, and I think she put this sentence in something that she's writing. You can almost consider the Earth to have been two different habitable planets so far, with two very different sets of conditions. And so this transition dramatically changed everything, right? But in particular, the chemistry of all redox sensitive elements and also the evolution of life. So we'll look a little bit at what the metals did. So here's just a repeat of the anoxic water column, the slightly oxidized water over sulfidic water, and then the modern water column, which is very well oxygenated except for this oxygen minimum zone here. If we put the metals on there, you'll see there are two axes, right? So iron and nickel are on the left and zinc, copper are on the right scale. But what you can see is that over that transition, iron and zinc and nickel and copper's solubility in the oceans went way down, sometimes by more than an order of magnitude, or at least an order of magnitude. And so that would have changed very much, right? All the organisms that were dependent on iron and zinc and nickel and copper in the ocean faced a huge crisis, and some went extinct, and some had to evolve a way to use some different metal, right? Or all kinds of evolutionary strategies, but also a change in what organisms were the most productive and most favored in the oceans. So what caused that big transition to happen? For oxygen to start permanently accumulating in the atmosphere, either there must have been an increased fluxes from things that produce oxygen and put it in the atmosphere, or decreased fluxes into sinks of oxygen, or both. So the dominant source of oxygen today, and probably always, is photosynthesis. And it's been a long-held consensus that oxygenic photosynthesis started at least 3 billion years ago. But in 2017, Woody Fisher at Caltech has renewed a debate over exactly when oxygenic photosynthesis started. There's a really nice review that came out very recently by Catling and Zonley to look at the arguments for old oxygenic photosynthesis versus having it evolve just at the start of the Great Oxidation event. And they seem to favor the former, that oxygenic photosynthesis was around for a while before oxygen could persistently accumulate in the atmosphere. Okay, how about the sinks? Where does oxygen get consumed? It gets consumed by weathering reactions. It gets consumed when organic matter piles up in the sediment, and then bacteria use the oxygen to help break down, decay the organic matter. And also oxygen reacts with methane to make CO2 and water. So these are the possible sources. What's pretty clear, Kevin Zonley and Mark Clare and David Catling have done some modeling studies where they've tried to figure out, with the best constraints they have on the fluxes from the sources and sinks, what would the atmosphere have looked like at different times? And there are lots of uncertainties in that, but one very robust conclusion that comes out of it is the Great Oxidation event could never have happened without some significant reduction in the amount of methane being put into the atmosphere. Also, there's quite a bit of evidence that most of the methane that was in the atmosphere would have been biogenic. It's being produced by methanogens, which are archaea. And so there's a little picture of some archaea cells, methanogens. And so these guys are producing methane, right, by metabolizing other forms of carbon. Okay, so they've done some modeling. Here's one of the recent papers showing that. So here on the y-axis is the concentrations, right, the fractions of free oxygen and methane in the atmosphere. And here's time on the bottom. So the Great Oxidation event happens around 2.40 billion years. And they tried lots and lots of different possibilities for the concentrations of these things. The only way they could get the model to do the Great Oxidation event, to allow the oxygen level in the atmosphere to rise, was to have methane go down by quite a bit. Here it's going down by about a factor of 6. Right? In any models where the methane didn't go down significantly, the great oxidation event could never happen. Right? You cannot have the O2 ramp up. So to tip the system in their model into a free oxygen bearing state, methane had to go down. But then one question is, why didn't it go down even more? Right? Here they're estimated it went down by a factor of 6. But what is it that controlled the minimum concentration of methane? And the reason that's so important is that if the atmosphere retained too much methane, the great oxidation event wouldn't have happened, right? Because the methane would react with the oxygen and cancel it out. Had the minimum gone too low, we removed so much methane, that was the primary greenhouse gas at the time. And the sun was fainter than it is now. And the planet, without enough methane to stay warm, would have turned into a prolonged icehouse or turned it into ice planet. So somehow all of the factors that affected the methane in the atmosphere and the oxygen somehow made it the case that the amount of methane was just right, like in the Goldilocks zone, to allow the great oxidation event to happen and to avoid the planet freezing over. Right? There were some short-lived freezing overs, or the first big glaciation events at that time, but the planet was not stuck in an ice state. So there are two hypotheses for what happened to the methanogens and why they stopped making so much methane. One of them is that the first pulses of oxygenation drove weathering of rocks, which allowed a lot of sulfate to come out of sulfide minerals and flow into the ocean, which stimulated the productivity of sulfate reducing bacteria. And there are a bunch of experiments showing that if you have sulfate reducing bacteria and methanogens in a neat little ecosystem, the sulfate reducing bacteria will outcompete methanogens for some major nutrients, right? So they'll be favored. So that's one thing that could have happened to drive methanogens down. But then another hypothesis got published in 2009 from Kurt Konhauser saying that these methanogens were in trouble well before any oxygen rose, and they suffered from a nickel famine. And that's because these methanogens must have nickel as the center. It's part of this cofactor in the enzyme that they're using for the final reduction step to make methane, right? So the methanogens cannot make methane, they cannot survive without having a fair amount of nickel around. And Konhauser thinks that the amount of nickel available to these organisms started to go down a few 100 million years before the great oxidation event happened. What makes him think that is this work that he did, where he measured in banded iron formations, and here's a picture of those. Just, you guys are in Minnesota, you know what this is. These stripy rocks with iron oxide and silica in alternating layers. So he took hundreds of samples of those and measured the molar nickel to iron ratio as a function of age. And the red points, the circles on this thing, the circles are whole rocks that he dissolved and measured. And then all the little plus symbols are laser ablation measurements that he made of the nickel iron ratio. And so you can see the first order pattern here that in the Archean, before 2.7 billion years ago, the amount of nickel could be pretty high, right? The highest values are seen in the Archean. And then between 2.7 and 2.4 billion years ago, the nickel level drops off significantly. And the great oxidation event happens after that, some 300 million years of nickel decline. And then the nickel level never goes back up to what it had been. Right? So this is his evidence to say that, that a nickel decline happened, which got the methanogens in trouble, dropped the methane in the atmosphere, and allowed oxygen to rise in the atmosphere. Okay, so the things that Konhauser is measuring here is the concentration or the ratio of nickel to iron in the rocks, the banded iron formations. And so then we really want to know how much nickel was in coexisting seawater. And so to figure that out, Konhauser did some experiments to learn if you simulate precipitation of banded iron formations, or the precursor phase for that, ferrihydrite, if you simulate that in the lab with some nickel in there, how much nickel gets into the ferrihydrite compared as a function of what's in the seawater. So here are the results of those experiments. Oops, what have I done? There we go. So on the x-axis is time, on the y-axis is their best estimate of how much nickel was in seawater in nanomoles per kilogram. And they did the experiments at pH 7, and they changed in their solutions the amount of dissolved silica, because the way that nickel partitions into the ferrihydride is a strong function of dissolved silica. It's now fairly widely held theory that the ocean was saturated or nearly saturated with silica. So that dark point at the top, 2.20 millimolars of silica, is probably the best estimate of what it was, which makes this dark gray hump on this plot probably the best estimate we have of how the nickel concentration went down from about 400 nanomolar to about 80 nanomolar after the rise of oxygen. And so it went down by about a factor of 5. Is a factor of 5 enough to make the methanogens really hurt for nickel? And so there's a compilation of culturing experiments with methanogens and the amount of nickel that they have available, and cyanobacteria. And this was compiled by Glass and DuPont. And their results show that the red squares mean that the element nickel is toxic. So cyanobacteria find these levels of nickel, or 10 to the -7, something times 10 to the -7, to be toxic. Well, the methanogens love that. Right? And then if the nickel level gets too low, methanogens are hurting for nickel, and cyanobacteria are much happier. So now we'll put on Konhauser's estimates that the Archean Ocean before 2.7 had 400 nanomolar of nickel. So at that condition, methanogens are quite happy. They have just the amount of nickel that they want, and they can be very productive. And meanwhile, cyanobacteria who coexisted with them were actually struggling with the too much nickel there. But once the nickel amount drops to about 80 nanomolars, now methanogens are limited for nickel. They don't have enough to be very productive, and the cyanobacteria are very happy and maybe positioned to outcompete methanogens, right, when the nickel had gone down. So the question then is, how do we find out whether methanogens really did decline as the nickel availability in seawater dropped? And that's a really hard thing to do because nothing about methanogens gets preserved in the rock record. So it's not clear, there's no way to go out and measure in rocks how abundant were methanogens at different times. Right? There's not a good record of that. You can play around with carbon isotopes, but it's extremely difficult to reach any conclusion. So I got to thinking about this in 2014, that maybe there would be some kind of stable isotope record of methanogen productivity in the past that we could find in the rocks, not the organisms, but a trace of their existence. So for those of you who don't think about isotopes very much, here's one example of how isotopes can fractionate, or the ratios of the heavy and light isotopes can vary when different chemical reactions happen. So one example is an equilibrium between two different phases. The isotopes of hydrogen and oxygen will exchange between ice and liquid water, so that the heavier isotopes end up in the ice and the lighter isotopes are in the water. Right? So the ratios of heavy to light in the ice and water are somewhat different. And then another example is that photosynthesis, or the fixation of carbon, fractionates carbon isotopes and oxygen isotopes. So if you have a pond full of algae, right, and there's the atmosphere, and you have the source of carbon, when the organisms take up a fraction of that carbon, they will preferentially take up the lighter isotope, carbon-12, and preferentially leave behind the heavier isotope. And so if that happens somewhere, and then the reservoir of carbon that was in the algae gets somehow physically separated from the reservoir that was in the water, that their activity, their presence will be recorded in the rock record. So the elements that can do this fractionation and can have biological fingerprints include the 5 that are highlighted right here, which I think people have now been measuring for about 70 years. And I will argue that there's almost no field at all in earth science that hasn't been profoundly enhanced by advances that are only possible through stable isotope work. If anyone wants to refute that, we can talk about it with the cookies and coffee at the end. All right, so now in the last 20 years or so, all of the highlighted elements shown now are known to have stable isotope fractionation in nature. So there's a ton of them, right? All these elements, the relative mass difference between the heavy and light isotopes for these are really small, but we now have instrumentation that can make very precise measurements of those isotope ratios. and can separate out and resolve differences between natural samples. So your faculty, Xinyuan Zheng and Larry Edwards, have a number of the instruments that can make these measurements, and a beautiful clean lab in which to prepare the samples for those analyses. So they know that the sample purification analysis is really, really hard. One of the things you have to do is purify from your sample the element that you want to measure. And so there's a bunch of hours standing in a funny suit in an all plastic room with HEPA filtered air to do the purification procedures. It's pretty tedious and it doesn't always work. One of the things that helps you cope with that is to make a 20:1 scale Lego model of the whole situation just for a good break. And then here's the instrument that makes the measurements. This thing's called a multiple collector inductively coupled plasma mass spectrometer. But what it does, at the front end, you ionize your entire sample, which is dissolved in a little bit of dilute nitric acid. And the ionized atoms in your sample get accelerated by a voltage down the flight tube of this instrument. And under this white cover, there's a very strong magnet that makes the charged particles turn a corner. And the light isotopes turn a sharp corner, and the heavier isotopes turn a less sharp corner, so that they are physically separated. And down here, some detectors can measure how many are arriving at the detector per unit time, so that you can get the ratio. And ours is called Kermit because we're Sesame Lab. Okay, so biology can sometimes leave behind some sort of isotopic fingerprint. If the productivity of a certain kind of organism goes up or down, that can be recorded in the rock record. So it's important to know also that methanogens do strongly fractionate the stable isotopes of nickel that was discovered 13 years ago at Penn State. And so maybe methanogens really declining, right, by a factor of 6 or something, left behind a nickel isotope fingerprint somewhere in the rock record, so that we could figure out if this decrease in nickel really did correspond with a decrease in methanogens. And so we're going to try to use the stable isotopes of nickel, there are 5 of them, ranging from 58 to 64, to look in the rock record to see if there's some shift associated with the decline in nickel concentration. And so I'll tell you a little bit about the notation for the isotope data. All the data are reported with the heavy to light 60:58 ratio relative to some standard chunk of nickel metal that we buy from NIST. And so then we talk about the isotope composition as being little delta 60:58, or the small change in the ratio of 60:58 compared to the sample, as just the relative difference in the ratio compared to the standard. And then the numbers come out to be really tiny fractions, because 60 and 58 are only 2 mass units apart, and this ratio is not going to vary terribly much. We multiply all those numbers by 1,000 and refer to the isotopic composition as being 0 per mil or 0.3 per mil. So our long-term internal precision is plus or minus 5/100th of a per mil. And if we get some rock standards and measure them over and over the long term, it's about plus or minus 0.09 per mil. And so far, the whole range of terrestrial samples that have been measured span about 3.5 per mil, right? Just to give you an idea of what variation is out there and what we can resolve. Okay, so here's that paper from 2009 showing that methanogens do fractionate nickel isotopes. And so Bill Cameron did this experiment. First, this gray bar represents a whole bunch of meteorites, basalts, granites, continental sediments, glacial lists. She took a whole bunch of different kinds of continental rocks, and they all fell in this very tight range between minus 0.1 and plus 0.1. So they're all about the same. Then she grew 3 species of methanogenic archaea, the 3 boxes on the left, and 1 species of archaea that does not make methane and doesn't care about nickel. And what she found in those experiments, these dark points are the initial composition, right, or the solution, and the methanogens themselves, she measured the nickel that they took up from the culture solution and found that they were lighter by an average of 1 per mil, right? So that in this case, it's most likely that they're taking up the light isotopes of nickel a little bit faster, because they have some protein on their membranes that take up the nickel, and the reaction to get the nickel atom in and take it up is just a little bit faster rate than the heavy isotopes. That's probably what's driving that effect. And so maybe if we measured nickel isotopes in the same banded iron formations that Kurt Konhauser was measuring, maybe we would be able to see some sort of shift in nickel isotopes as the nickel was falling in concentration. And so one way that this could work, here's a plot that shows the fraction of the nickel available that is getting taken up by the methanogenic biomass. Right? So it starts at 0 and gets to, they're taking up all the nickel that's available. And then the y-axis is isotopic composition. Here's the composition of modern seawater at 1.4 per mil. And compared to that, methanogens would fractionate by 1 per mil. So their composition down here would be at plus 0.4 or something. If you then had an increase in methanogen productivity such that the biomass is taking up a greater and greater fraction of the nickel that's there, the water's composition would evolve along this solid blue line, and the methanogen's biomass nickel composition would evolve along this dashed green line. And so eventually, if they become really limited for nickel and they're taking up most of what's there, the seawater should become heavier isotopically. And then all other types of marine sediment that precipitate from ocean water may reflect that. Right? This is how maybe there would be some biosignature recorded. And there's a little thing just to show you our 2 sigma, 2 standard deviation uncertainty, so that you can see how much would the fraction of nickel being taken up by the biomass have to change in order for us to see a difference in the isotopes, right? It's not all that terribly much. Okay, so does this work? We measured all the banded iron formations and we're looking to see if the delta nickel values, delta 60 values get higher as nickel concentration drops from 2.7 to 2.4. And there are the results. So that's not what we expected. In the Archean, before the nickel dropped, all the values for the banded iron formations fall in this fairly tight range. But then starting at 2.7, this pink box right here is the interval where Konhauser saw nickel concentration dropping. The nickel isotope values are all over the place. Some of them are much heavier than they were before, and some of them are much lighter. So it's not what we were kind of hoping to just see an increase here, but we don't. And then as you can see, the few intervals of time where there still is anything like a banded iron formation, the values are all over the place for a big spread, which I am not going to be able to explain to you today. Okay, so it turns out that that whole idea of methanogens leaving a biosignature is really stupid. But I sent the idea to NSF and to NASA in 2014, and all those reviewers didn't figure out that this was actually not a very good idea at all. And the reason is that for this to have happened, for methanogens to really have started taking up so much nickel that we could see a difference in the whole ocean's nickel isotopes, the oceans would have had to be a couple percent methanogens by mass, which would be a very thick soup of methanogens, which is utterly unreasonable. So it turns out that whole approach was actually not a very good idea, to try to consider the ocean as one big bucket with Rayleigh fractionation of nickel. So that problem, right, if there is a way to see a direct biological fingerprint of methanogens, it's going to be much harder than that. So what we would have to do instead is find a way to measure the burial flux of methanogen nickel. As is done for carbon isotopes, we measure the fluxes of carbon into organic rich sediments and into carbonate sediments. And the way we do that is by knowing how isotopes fractionate from seawater into carbonate sediments and organic matter sediments, and then analyzing multiple rock types that have the same over the same time intervals, and reconstructing how the proportions of the carbonate sink for carbon and the organic sink for carbon have shifted over time. Right, so that's been an amazing set of advances to figure out how the organic matter burial has varied over time. Maybe something like that's possible for nickel, but it's going to be hard. Check with me in 5 years. Okay, so meanwhile, what can we learn from the banded iron formation data? So I've put the same data on the plot here, but what I've done is adjusted all the points, because what we measured was the rocks, the banded iron formation, which is the nickel that precipitated with iron oxides in the first place. And what I really want to be looking at is nickel isotopes of seawater. And so my postdoc and I have another paper, and I won't show you the details today, showing that there's a 0.3 per mil offset, that the nickel that gets precipitated with iron oxides is 3 per mil, 0.3 per mil lighter than what's in the coexisting solution. So here I've just shifted all of the banded iron formation points up on this plot by 0.3 per mil to show an estimate of what seawater nickel isotopes would have looked like that coexisted with each of those BIF samples. And so the first thing we can see is that modern seawater is plus 1.4, but the Archean seawater was more like 0.8. And so the Archean ocean was lighter in nickel isotopes than it is now. And the best hypothesis we can come up to explain that is that now oxidizing weathering, chemical weathering happens to exposed rocks all over the surface of the earth now. And the lighter nickel isotopes are retained by the formation of iron oxides on land, and the heavier dissolved nickel is what flows to the ocean. And so that's what makes the ocean heavy now. Of course, oxidative weathering wasn't a thing in the Archean. And so the light nickel would have just gone to the ocean with the heavy nickel, making the ocean somewhat lighter. The second thing we can see is that between 2.8 and 2.1 billion, the values are over a huge spread. Some values are much lighter or much heavier than the modern ocean. Something could have made the ocean even heavier than it is now. And sometimes the ocean was so light that it was even lighter than bulk silicate Earth. Right? So it's very interesting to ponder what are all the possible ways that you could have generated in that time interval banded iron formations and seawater that were extremely heavy and extremely light. And so what are some of those processes? A smaller nickel reservoir, right? If the nickel concentration went down by a factor of 5, the reservoir is smaller. And so any changes in the sources or the sinks would swing the isotopic composition of seawater more strongly if there's just less of it there. But if you have an enormous reservoir of nickel in the ocean, then it's really hard to perturb its isotope composition. So it might be the reason that this big spread is there is in part because the reservoir was smaller. So small perturbations show up more. To make the heavier compositions, we do know that there was more sulfate, and the bacterial sulfate reducers made sulfide. And so nickel could precipitate in sulfides, and those are isotopically light. So that would have left remaining seawater heavy. I can't really think of very good processes that could have driven seawater to be lighter than bulk silicate Earth. So I have to just leave that one there right now. My former postdoc and I, Shuijong and I, are actually kind of in an argument right now about this paper. We're trying to write up this paper and figure out how we're going to explain all that. So check with me in 6 months. All right. So one of the questions that we thought of, though, while we were pondering this plot, did the amount or the isotopic of nickel that was coming from continental weathering change somehow from the Archean to the Proterozoic? And so to examine that question, we'll first look at this. What did the nickel concentration do in the upper continental crust that is the source of nickel that gets weathered out and flows to the ocean? So Brendan Keller and Schurner wrote this paper 10 years ago. They analyzed more than 70,000 samples of continental igneous rocks of different ages, which is an astounding feat. That's a lot of lab work and analysis. And they came to the conclusion right here, you can see for a whole bunch of elements, but in particular for nickel, that this drop in nickel concentration that Konhauser sees in marine sediments also shows up in how much nickel is in continental igneous rocks over time, right? So here's this decrease. And so the possible thing is that because the mantle was, the whole Earth is cooling, that when you make magmas with a high mantle potential temperature, you make melts of higher fractions of the mantle. A lot of olivine is melting, and olivine hosts a lot of nickel. So early in Earth history, when you're making those higher fraction melts, those would be nickel-rich melts and would make a nickel-rich continental crust. Later on, when the mantle is cooling off and you're melting the mantle at lower potential temperatures, you would be not putting so much olivine into the magma. And so the nickel concentration of the igneous rocks would be lower. And so it probably is the case that by the time we were really forming thicker continents, somewhere around 2.9 billion, that there was a growing proportion of nickel poor magmas, and then recycling of the crust and further differentiation of the igneous rocks would have led to felsic rocks being exposed, which are very low in nickel. So maybe that's the whole story. that the nickel concentration, the crust went down, so there was just less going to the ocean, and that's it. But what we did to look at this is we analyzed some diamictites. And what these things are basically the chemical weathering residue that's shoved around by a glacier and turned into a sedimentary rock. So what we're measuring is a sample, and these things are composites on all different continents of many, many samples of this glacial rock, this glacial diamictite. And so we measured the nickel concentration in those and found that the nickel concentration is indeed higher at 2.9 billion years and lower at 2.4, and then it never rises, right? So it just looks just like the pattern that Konhauser saw in the banded iron formations and reflects this decrease of nickel in continental igneous rocks. And the nickel concentration drops by about a factor of 3, right? So maybe that's all there is to this. That might have been the case, but of course we measured the nickel isotopes of these diamictites and learned something very interesting. So here's the same plot I just showed you on the upper right, showing the nickel concentration is going down. But here's the isotope composition of these diamictites, and it's lighter at 2.9 and significantly heavier at 2.4. If you just had the continental crust having less and less nickel in it, you wouldn't expect to see a change in the isotopes in the stuff that glaciers shove around and turn into sediments. That's sort of a mixed up average of what the continental crust might look like, right? So we can't explain this, right, by crustal evolution over time, that there's just less nickel rich mafic rock and more felsic rock. And the reason is that all these igneous rocks, komatiites, basalts, TTG suite rocks from the Archean, granites, right, these are the things that Vil Cameron measured. They're all around 0.2 or 0.1 per mil, as is the mantle and the best estimate of bulk silicate earth. So even if you change the proportions of komatiites, basalts, TTG suite, felsic rocks, you can't account for the shift in the isotopes of the diamictites that way, except that sulfide minerals and nickel sulfide ores are the only thing that are really, really isotopically light, right? So here's where the isotope contrast can come from, that you could be weathering different amounts of sulfide at different times, right? And the diamictites, it's important to remember that you have a crust, some chemical weathering happens and some metal is dissolved out of the crust and that goes to the ocean. And there's a chemical weathering residue left that the glaciers can then scrape and mix around and turn into a diamictite that we can analyze billions of years later, right? So our hypothesis now is that the onset of sulfide weathering a little bit before the great oxidation event happened allowed light nickel to weather out of the sulfides and go to the ocean, making the ocean lighter, but also made the upper continental crust heavier because the light isotopes left and went to the ocean, and the heavier isotopes remained in the continental crust. Right, so here's the same data again, just to show you that all these things line up. The total sulfur content of the diamictites goes down, which is consistent with sulfide minerals weathering out more. The nickel goes down about threefold, right? So it looks like all of these things are lining up to point towards sulfide weathering starting about 2.7 billion years ago. And there are lots of papers looking at other elements that are hosted in sulfides and showing this same trend, that it looks like sulfides really started to weather pretty considerably a little bit before the great oxidation event, a little bit being 300 million years. So what we wanted to do next then, to add something to that idea that sulfide weathering started, was to attempt to quantify how much did the fraction of nickel coming out of sulfide minerals versus silicate minerals change before versus after the great oxidation event. And so here we made this little mass balance model, and we said that in the continental crust, here's a pie chart showing all of the continental crust, that there's some fraction of the nickel in it that will be chemically weathered and will be dissolved and will flow to the ocean. And then the remainder, or 1 minus F, most of it, is nickel that doesn't chemically weather and it stays in the crust. And so we can write some mass balance equations, one of them for before the GOE at 2.9, or even before the nickel decline, that says the fraction of the nickel that weathered out of the crust times the isotopic composition of that nickel, plus the rest that didn't get weathered times the nickel isotopic composition of the rest, equals what the initial crust was, right? So the parts equal the sum of the parts is the whole. And then a second equation for a different time interval after the great oxidation event, where the amount that weathered might be different. And we do know already, because we measured it, that the diamictites got heavier. Right? So then of the fraction that weathers, part of that is coming out of sulfide minerals, and the rest of it is weathering out of silicate minerals. So we can write another mass balance equation that goes with this little pie, right? That the fraction of nickel that's coming from sulfides times the isotopic composition of those sulfides, plus the complement of that times the isotopic composition of silicate rocks, equals what weathered, right? And another equation just like that for the second time period. So remember, our question up here is, can we quantify the increase in the fraction of nickel that's derived from sulfide weathering? Right. So what I'm interested in knowing is the fraction of nickel coming from sulfides in one time period versus the other. What's the difference in the fraction of nickel being weathered out of sulfides versus silicates before and after? So when we put some plausible values in for how much nickel total would have weathered out of the crust, and do some calculations using the initial composition of the crust and reflecting the uncertainties with this little family of curves, the answer, right, the fraction of nickel coming from sulfides should lie on one of these curves. And because nickel's not very soluble, we're just going to guess or assert that the fraction of nickel that comes out of the crust that dissolves it all is 10% or smaller, right, 0.1 or smaller. And so the values that work for the change in the fraction coming from sulfides are in this red box. So at least 30% increase in the fraction of nickel that was coming out of sulfides, if the overall weathering intensity of the crust hadn't changed, right? So we also tried some different values for how much increase in weathering intensity might there have been due to the rise of oxygen. And so we have another family of curves here with slightly different values. And the values of delta F that work are 40% and bigger. Right? So the most conservative case that we can make shows that the fraction of sulfide-derived nickel during weathering must have increased at least 30% from before until after the fall of nickel concentration and the rise of oxygen. That's a lot, right? So here's our first constraint on how much did sulfide weathering turn on, and how much did sulfide become the source of nickel and other trace metals that are flowing to the ocean. Okay, so it turns out that this sulfide weathering thing's a double-edged sword for these late Archean methanogens. There was more sulfate going to the ocean because of all the sulfide weathering, but that stimulated the competitors, right? Fueled their competitors, so that was bad for them. But at the same time, if sulfides had not started to release significant nickel, they would have starved even worse for nickel, right? The seawater nickel would have plummeted, and that maybe would have been a much, much worse situation for methanogens, right? So it's sort of trade-offs for them. And had that happened, if the methanogen had really been knocked back very severely, maybe the fate of the Earth would have been different. And right now, of course, you guys are in Minneapolis, so you are standing on a frozen planet, but maybe the whole thing would have been frozen forever. A very different planetary outcome than we have. Okay, so just to summarize, we set out at the beginning to figure out whether banded iron formations would tell us about a methanogen decline, but the results are pretty hard to interpret, and it probably isn't, we're not going to see a direct biological fingerprint in that rock. However, we can see that the nickel cycle in the ocean changed from something that was pretty constant, pretty tightly regulated maybe, and isotopically consistent, to something that's wildly fluctuating, right? Some sort of nickel cycle that's much more susceptible to perturbations and changes in the types of marine sedimentation and different changes in what minerals were precipitating and where would have swung the ocean's isotopic composition around. Right? So in these glacial sediments, we can learn one more thing, which is that we see the isotopes changing in those glacial chemical weathering residues, and that we can't explain it by just a change in the proportions of ultramafic, mafic, and felsic rocks, because that wouldn't change the isotopes. Right? We have to invoke the weathering of sulfide minerals to make that change. And from that, we can try to make a constraint and learn that the sulfide fraction of nickel increased by a lot, 30% or more. Right? And so the onset of sulfide weathering released just enough nickel, right, to keep methane production in the Goldilocks zone. Too much methane, there never would have been a rise of oxygen. Too little methane, the planet could have frozen. Right? So it's sort of lucky or unlucky, depending on your viewpoint, that it all unfolded just as it did. All right, so with that all, thank you very much for your attention. You can send me any questions by email, or maybe we'll talk for just a few minutes here or use the chat window. If you have a chance, come visit us in Flagstaff. We don't have very many lakes, but we have sticking up topography and sticking down topography, which is pretty exciting. All right, so thank you very much.
All right, thank you, Laura. Laura, our tradition is that we give the opportunity to questions first to graduate students. So I see a few hands up, but not the faculty yet. So those, Nicole, I guess, is the one I see. So go ahead.
Oh, sorry, I was just applauding. I
Oh,
love.
okay.
Clapping hands
I
in
misunderstood. Okay, but any graduate students have the opportunity now for students before the faculty or others jump in. Oh, okay, in the chat, I will read it. Great talk. I'm a big fan of Kermit. I liked hearing about the way the project progressed.
Hi, Laura. This
Hi.
is Untitled. I'm also an experimental petrologist as of right now.
Wait, is there should I be reading something between the lines?
No. So I really enjoyed your talk, and I know nothing about biogeochemistry, but how can we say that methanogens fractionate nickel the same way they do now with like the experiments that people do compared to how they used to fractionate nickel in the Archean?
The reason we can say that is that at the moment there is exactly one paper that did the experiments with modern strains of methanogens and measured that fractionation. And that's all we have. And so that's what we're going with for now. Right there. To my knowledge, no one else has followed up by looking at different methanogen species, more of them, or changing conditions, or thinking about trying to figure out what genetic differences there might have been in the ancient past with phylogenetic approaches to figure out if it's likely that that fractionation has always been the same or not. Of course, as Mark, does your email signature still say the simple things you see are all complicated or something like that? Yeah.
I removed that from my email at some point, but it did for many years, right?
Yeah, so this is one of those things where it's simple because there's just one paper. And as soon as anyone else tries to work on this or push it ahead, there will be some complications, right? I would be surprised if it turns out that all methanogens now, under all conditions, and anything we can learn about past methanogens would all make the same fractionation, right? That seems pretty unlikely. But I don't know, that's kind of all we have to go with at the moment. So we're trying to just be on the cutting edge with what we have available.
Thank you so much.
I can't recommend growing methanogens.
Any other questions from graduate students before we open it up? Okay, I think I saw what, Jake and Vera's hands up, or maybe you guys were just applauding. I don't know. Was that a question, Jake?
Yes, Laura, I enjoyed your talk. One thing I was wondering about is it seems to me like the concentration of nickel in the modern ocean must even be lower than it was in the Archean. But are there examples of environments today where nickel is limiting for methanogens? To me, it seems like the places what they're limited by is actually organic substrates. So wherever we have a lot of organic matter, like rice paddy or shelf or something like that, then you have a lot of methanogens. And I wonder if those are actually limited by nickel.
So yeah, so I think everything you said is just right. So today, methanogens are not like a big dominant form of life like they were in the Archean, right? Today in the ocean, they're sort of relegated to in sediments that are pretty reducing. And I think you're right that nickel is very unlikely to be the limiting factor for them, that it probably is just organic matter to break down as the source of carbon. So I don't think nickel limitation in the oceans is a thing now. As far as rice paddies or wetlands or things like highly reducing conditions on land, I don't know. So if nickel is ever the limiting factor, probably my guess is maybe not, right? But back in the Archean, there were so many methanogens and they must have been pumping out such a huge flux that the amount of nickel available could well have been a limiting factor.
Okay, thank you.
Okay,
Kara?
there's Jeff. Oh,
Oh.
go ahead.
Go ahead, Kara.
Okay, sorry. Thanks for that talk, Laura. It was really great. Okay, this might show my ignorance of isotope systems, but how much does nickel speciation matter for the isotope systems? And I'm thinking about it, especially in relation to Jake's last question about the concentration, because nickel speciation is going to be controlled so much by the pH as well as the concentration. Like there's so many factors that influence it. And so I was wondering how that would influence like your interpretation of the data and how much it causes fractionation. And yeah. So
I think you're asking, there's the speciation of nickel in today's ocean at pH 8.2, 8.19, 8.18, 8.17. We're gonna have to keep up with the change. But there's the speciation that's in the modern ocean, but the Archean ocean was probably different. The pH was almost certainly lower. Last night, after talking with Bill Seafried yesterday, I started poking around and found some people estimating that was about 7 in the Archean ocean. So that like the total carbonate also would matter. But what really matters is the pH, because you do make some nickel hydroxide species and a little bit of nickel chloride. And those things fractionate a little bit, like on the order of a tenth of a per mil from just nickel 2+ free ion. So there probably is, if we could really resolve all these things and pin everything down really precisely, we probably would be able to see the effect of different pH and different speciation. Obviously, once sulfide's in the water, you start having nickel sulfide aqueous species also. But those fractionations, there's a theoretical paper calculating with density functional theory what those fractionations between nickel species are, and none of those are all that big, except for if you stick nickel in some big crown ether thing, right? And I keep wondering if nickel in porphyrin structures, right, which is how they are in enzymes, if those would be strongly fractionated from free nickel, but no one's attempted a measurement yet.
Okay, great, thanks a lot.
Yep.
There's a question in the chat that I'll read before Xinwan. So Jeff Havig says, great talk, Laura. I was curious. Oops, wait a minute. I was curious if you had thought at all about nickel-containing enzymes. Methanogens always get all the hype. For example, hydrogenase and hydrogen metabolisms might have been important with serpentinization and fermentation generating H2.
Right. So Jeff is right. There are other enzymes and other organisms that have biological demand for nickel. Methanogens are the winner. Right? The amount of nickel they need per mole of carbon reduced is much bigger than the amount of nickel that these other hydrogenases or other enzymes, other organisms need. But I think you're probably right that I don't know enough about serpentinization and fermentation that makes H2. But it would shock me if the organisms doing that don't also fractionate nickel isotopes and probably also preferentially take up the lighter isotopes. And so then the question would be, can you find a system where there's enough of that going on, enough fractionation, and then a mechanism to physically separate the fractionated reservoirs from each other such that you could find them, actually resolve that or measure it. And then the next question, if they fractionate and you can resolve it, can you then find the science question that fractionation would answer, right? And I have no idea, and I don't think anyone's really tried. There are so far 0 methods of hydrothermal fluid and almost nothing whatsoever on oceanic crust that's altered in any way, right? So I don't know, sometimes I think about trying to do experiments or trying to get samples of altered oceanic crust that are not horribly complicated, just try to take a first order look at that. But so far, no work has been done to look at that.
Xinwan?
Hi, Laura. That's very nice talk. I'm just curious, does the nickel isotope fractionation vary with the amount of silicon?
I don't know.
Yeah.
Yeah, the experiments that Chuijong and I did, had no silica, no dissolved silica.
Mhm.
Right. But I think so the more silica you add, I think the less nickel partitions into the iron oxides. But
Right.
whether the nickel isotope effect is different, nobody knows right now.
Mhm. Have you tried to explore your nickel isotope data versus maybe silicon iron ratio in the beef samples you analyzed?
That's a good question, right? To see if the nickel isotopes. So I asked Shuijong to look, do those nickel isotopes correlate with anything in that huge set of banded iron formations? So he looked at the nickel isotopes against everything's concentration of, I don't know, 30 different elements, and nothing correlates very well at all. But I don't know that he specifically looked at the silicon iron ratio,
Okay.
which probably we should do.
Yeah. Well, I'm just curious. I know nothing about the nickel isotope fractionation, but clearly the silicon has an influence on the partition coefficient for the nickel incorporated into the iron.
Yeah. Okay, so my PhD student is on the Zoom, and so she just put a note in here. There's a new paper that she's read that I have not, which is that there are a paper looking at the effect of dissolved silica on nickel isotopes in iron oxyhydroxides, and apparently there's not a big difference.
Okay, that addressed my question.
Okay, thank you, Eva.
All right, thank you.
Yep, and there's one more question in the chat. Maybe methanogens could be used to consume and transform methane hydrates before they release the methane hydrate to the atmosphere. I've read that, but so far there doesn't seem to be a way to encourage this in a practical way. How common are methanogens in the ocean? They're really uncommon. There just isn't that much at all, because you need to have some sediments where there's just so many microbes, trying to oxidize the next thing and oxidize the next thing. And so that you have to get to extremely reducing conditions before methanogens can really be there and compete for and do carbon fixation there. I don't know if methanogens can do anything with methane hydrate. Personally, the idea of releasing methane hydrate is terrifying. Right? So I think maybe we're all better off if it never happens. But I don't know, it will happen. I have no idea what the interaction would be between methanogens and methane hydrates. Yeah, right. They don't eat methane, they make it. So I think they would maybe just leave the methane hydrates alone. Yeah, of course, it's going to happen as things warm up. What will the next?
I'll give the opportunity for one more question, and if there isn't one, I'll ask one. But to shut me up, you can ask your own question. Someone asks, what happened to the rise of nickel concentration in the igneous rocks around 1.5 GA?
So I don't know, but I mean, one thing that could do that, let me share one more time so everybody can see what the question is about. It's this guy right here. That at 1.5, of course, the error bar is so big, right? The range of values is so big that maybe it didn't really go up, or maybe it went up quite a lot at 1.5, 1.4. So I guess one way to do that is to make a whole pile of mafic rocks and put them in the crust at that time. Because you need to think about supercontinent cycles of assembly and breakup to see if there's like a big pulse of making and emplacing and exposing then more mafic rock. But at the moment, I can't name what tectonic events were going on that could have done that right there. Maybe someone else can drop that in the chat.
Well, I lied. I'm going to ask one last question. Laura, the fact that the oceans are now heavy in nickel, does that have to be a remnant of something in the past, or is there any way to explain that with the modern oceans? Or with modern nickel cycle?
NSF just is giving me a chunk of money right now to try to figure out why the hell is it so heavy now. I don't know the answer to that question, right? So the residence time of nickel in the ocean is like 10 or 20,000 years. So I think on really long time scales, it can't be a remnant of, what it has to be a function of is the sources to the ocean and their isotopic compositions, and the sink fluxes and their isotopic compositions. And if the ocean is at steady state over a long time, those must balance somehow. And there's no evidence from the rock record that there's been a big monotonic trend in one direction or another since the Permian-Triassic boundary, at least. So somehow the cycle appears to be near or at steady state, and it's got to be a function of the sources and the sinks, some of which we don't know. But I don't think it's a remnant of anything in the really deep past. No, yes, it is. The formation of Earth and the inventory of nickel atoms that the entire silicate Earth has. How about that?
Oh, but it could be a remnant of the past just in terms of the mean age of continental weathering or something like that.
Yeah.
All right.
Yeah, I don't know.
Laura, thanks. That was great. People can clap and cheer audibly or visibly, however they care. And if anybody has any further questions, I'm sure Laura would be happy to correspond via email.
I have some goofy stories about Mark from 30 years ago.
No, this is where we're going to stop the Zoom right now.
Yeah, but I think that's a two-way street.
Thanks, Laura. This was
Yeah.
great.
Thank you very much, everybody.
Thanks, Laura. It was so good
Yeah.
to see you.
I'll sign off and, Kara, you and Laura can talk after I'm gone.
We talked for like
Maybe-
an hour yesterday. We're all
Stay here.
mostly caught up. I don't know.
Yeah, but I didn't tell you the crazy stories. Yeah,
No,
I think
you
Mark-
Bye.
I think Mark has just as many.
All right. Thanks so much, Laura. It was so good to see you.
Yep, good to
It was
see
a
you
great
too.
talk. I learned a lot.
That's good. Yeah.
Thanks.
All right.
Bye.
Bye.